Yeah, thank you everybody for joining. This is Women in HPC at SDSC Talk Series, and I'm one of the co-founders of Women in HPC, and I have here Claire Sturm here with me and other Women in HPC committee leads like Cindy Avery and others. And I just want to briefly introduce what this chapter is about. Women in HPC at SDSC is our Southern California chapter. But Women in HPC by itself is a global organization. It's headquartered in United Kingdom. And our goal is to provide a platform, a space for women-identified professionals and allies to come share their career experience and support students and early career professionals to attend conferences. And we also provide mentorship assistance to students and faculty. So essentially, it's a space where early career professionals or students can come in and they share their stories and they can help each other in the process. We connect with other similar organizations like Society for Women Engineers, and if there are other organizations that you guys know of that we should connect with, please send us a note. Cindy, can you advance to the next slide, please? And here's a link to join our Google group. We have the talk series, and we also have some informal events in San Diego and that you can attend. It's more of a quarterly activity. And then we also do workshops and at major conferences and that, in case if you're all traveling, you could also attend. So with that, I think let me introduce our speaker, Dr. Anita Bandrowski. I've known Dr. Anita Bandrowski for many years now. She wears multiple hats. She's the CEO and founder of SciCrunch, a company, and also a project lead at the Center for Research for Biological Systems at UCSD. She received a PhD in psychology from UC Riverside and a postdoc from Stanford University in neurology. So please join me in welcoming Anita Bandrowski today.
Thank you so much. I need to, here we go. All right, I'm gonna share a really silly little slide here. It just has my name on it. Are you guys seeing that?
Yes.
Yes. Okay, awesome. And one of the things that it says on there is that I lead a company. I also lead I'm a principal investigator at what used to be CRBS. Thanks, Suba, I probably should have read that part. But we've renamed it into the Federate Informatics Lab at UCSD. And more recently, I actually am the co-founder and lead for something for a new nonprofit organization called rids.org. And we're just kind of starting to get that off the ground. And I'll show you why we care about that, hopefully, over the next few slides. And by the way, please feel free to, if you have any questions, just ask me, because I didn't prepare a lot of, this isn't really a formal talk. It's the meandering of my life talk. And if you have any questions or thoughts or comments, just, just shout them out. I can stop at any point. So in terms of, what's on my CV, you'll see things like, in 2000, I got a PhD in psychology. Strangely enough, we didn't actually have a neuroscience program at that time. And I was in an electrophysiology laboratory, which, is not what you think of when you think of psychologists. I did a postdoc at Stanford. I was also doing electrophysiology. This time I was studying epilepsy and not learning and memory. Right after that. And so this is a lovely picture of our neurology softball team, which I was a manager during postdoc. That's very important for the rest of it. No, it's not important for anything, but we were called the myoclonic jerks. And so I just thought, when I ran across this picture, I thought, I need to get the jerks back in there. So this was great. Our team pitcher is actually the CEO of a company, actually several companies. Our first baseman, who is not in this picture, was a pediatric neurologist. He was really tall. It was really great. And then we actually, for a while, we had a ringer because somebody went to med school, but they were getting recruited out by a professional baseball team. So we were really good for about 5 minutes. The rest of us weren't, but one of us was, and that helped. And then what happens with women sometimes is they tend to have things like this. And this one is now a bit bigger than this, but that happened after I went to after my postdoc. And so what happened with this is that I ended up actually leaving academia and went to work for a guy named Craig Venter, who kind of looks like this baby, but not exactly, because with Winston Churchill kind of a feel to him. But in 2005, I actually went. This was right around the time when we were getting to the point where we had the genome mostly assembled, and then they were asking questions. What do you do with all of this information? And there was this weird thing that no one has ever heard of. It was called curation. I think curation is still something that nobody else really knows about, and data curation is incredibly important. It turns out that when you look at data science tasks, about 80% of the time you're doing a task, you're not doing the machine learning, the statistics, all of this stuff, you're actually just cleaning data. And that is all curation stuff. So I got my feet wet with a lot of curation stuff in the company called Celera. In 2006, then, I did the standard, let's try to see if I can make it as a professor thing. As a teaching faculty, I didn't like that. So I went back into research. And so in 2008, I actually came to work at UCSD in a project called the Neuroscience Information Framework, which was a contract, something that I had not really experienced before, but I'll let you know how that's, why that's important in a little bit. And then a few years later, I again got very antsy and I went to work for Craig Venter again. That's why there are two pictures of him on this particular timeline. This time it was a company called Human Longevity, and I was one of the people tapped to put together a high throughput curation pipeline for the genomics data that he was creating for both insurance companies and a bunch of celebrities that would kind of rotate through. And then after that, I started with the RIDs, and there are a couple of different places where I went from UCSD back out to industry, back into UCSD, back into industry, and now I've got a foot in both, which is very weird. All right. So the goal of my life, at least since 2008, because everything before that was pretty pro forma, right? But it has been all about RIDs. And why are RIDs even a thing? Why do they exist? Why are we here? So in 2008, I told you that we had a contract with the National Institutes of Health. And that contract was to gather up a bunch of data and to make it all available to people who, would care about neuroscience data. I was tapped to help and figure this out. I was curating. Actually, I was the head of curation for this project when I first came in. And but yet there was this contract thing. And a contract, unlike a grant, in a grant, you can say, we're going to cure cancer, just give us some money. And they'll kind of give you money, and then you won't cure cancer. And then you'll say, hey, we're going to cure cancer with the next grant. And, they'll give you some money and you won't cure cancer. But a contract is different. Contract is like in, line 41, subsection B, there's a line here and you need to account for it. That's a very different way of actually doing science and thinking about stuff. So one of the things that was in one of those subsections was kind of, represented here. Not the exact language, obviously, but, NIH basically said, hey, people are wasting a lot of time looking for these things called antibodies in the scientific literature. Why can't you help them by creating an AI algorithm that can find those things so they don't have to waste their time? I mean, their time is valuable. I understand, right? So we actually had to do this, which was back in 2008. Like, we're pretty far before ChatGPT. And at the same time, or a little bit later, we started to find things out about antibodies and the fact that actually they are responsible for a whole lot of things, including the reproducibility crisis. There's a lot of problems with antibodies. And because this is an HPC kind of audience, I thought I would tell you what the heck an antibody is, because you might not be familiar. So actually, show of hands, how many people know what antibodies are? Woohoo! Okay, at least several people. And I think I saw Leo on here too. And I would imagine that he also knows what antibodies are, which is great. But a lot of people don't. And so what's an antibody? Well, essentially what you do is you get a protein or a thing that you care about, and you inject that into some kind of an animal. One of the things that may be injected or breathed in that you may have heard something about recently is this thing called COVID, right? And so that is the challenge to the immune system that comes in from outside. And generally when you generate antibodies, you do that by injecting it in blood, but you don't have to. You can also let the animal or the human breathe something in. And what happens in the immune system is there are these cells inside of basically the blood, right? That start to create little molecules that can actually bind to that thing, that challenge thing. And what happens with antibody production is you pull out the blood, you find some of these cells that are going to find that thing that you care about. You merge them with a cancer cell, and then you actually can, pull that out and pull out just the cells that are actually expressing the antibody that you want, that has a high affinity for the thing that you looked at before. For the protein that you care about. And then you can overexpress those, and then you can pull out these antibodies, and you can make people buy them. Well, you can't make people buy them, but you can offer them for sale. How's that? And so, kind of fun things about antibodies. Serum. So this is basically the blood of a horse, which was spun down very rudimentary ways, became the very first biologic treatment. It was well over 100 years ago, and that was antivenom. And it's still used as antivenom today, although it's a very dirty drug because there's lots of other antibodies that are in that horse serum that will then give large immune reactions when you get that antivenom. So there are some companies that are trying to do better. So this affinity purified serum, which is the what's referred to as the polyclonal antibody, is actually still about 50% of the total market of antibodies, and that's a $3 billion market. Some of the weird things about antibodies is that these cultures can be contaminated, and they can also experience genetic drift because they are living things, and living things tend to evolve. So who cares about antibodies? Like, yeah, we make them in our bodies, we can buy them, but who cares about these things? Well, I pulled the numbers from 2023, and these are kind of pretty. So there's something called a small molecule drug. And I think everybody has heard of small molecule drugs, right? Everybody's taken an aspirin. That's a small molecule. So small molecule drugs used to be 100% of the drug market, but now they're getting to be about 50% of the drug market. And if you look at the top 10 highest grossing drugs, so most popular, these are $10 billion or more kind of drugs per year. 70% are actually called biologics. And biologics are largely antibodies. So in 2001, 2002, they were largely monoclonal antibodies. And then now they're getting to be more recombinant. There are portions of antibodies that are being used. They're also called biologics, but these are essentially antibody-derived proteins. And if you look at for example, the autoimmune market, this is almost all antibody-based now. Oncology is still mostly small molecule, but that's also changing. By the way, that 46% for oncology, those are all of your new type drugs that are much more specific towards the oncology activity. And they are actually have many fewer side effects. So that's why people care about antibodies. So it's not just that these are some molecules that, some scientists in some basement use. These are actually really important. And so we asked, when we were asked by NIH about these antibodies and how to text mine these things out of the papers and create this magical AI algorithm that would actually pull these things out. We started doing curation. We looked at the papers and how these antibodies are actually being described. And so here's a lovely sentence that says, briefly, monolayers were permeabilized and fixed using 100% methanol held at minus 20 C for 5 minutes and then blocked and labeled with mouse anti-flag from Sigma. At this dilution, blah, 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 and rabbit anti-MIC from Protein Tech. Okay, cool. So this is an antibody, mouse anti-flag from Sigma, right? This is all that you get out of this paper about this reagent. And when you go to Sigma, first of all, you'll notice something really quickly, is that it's not just Sigma, it's millipore Sigma. And you'll see that there is 17 different antibodies for this thing here. And so if we were to magically come up with an antibody detector or finder or suggester algorithm, then we would be having to look up things like this and pretend that we had some idea about what the author meant. So this is still a problem, by the way, in the scientific literature. If you look at this particular antibody from a 2025 paper, there's a whole bunch of products that actually match that particular antibody, even though, there look like there's some numbers in there. Okay. So what we did is we failed completely and utterly, and we told NIH, hey guys, we have no idea how to create an algorithm. We failed to find the basic information that needs to be there in a paper. We can come up with an algorithm, but it'll be trash because it'll be guessing. And scientists, I don't know about you all, but we don't like to guess. We don't like to make stuff up, which is something that you may or may not have heard about. Algorithms are able to do very well. So yes, could we have created an algorithm at that point? Sure. Would it have been trash? Yes, it would. So what we did instead is we created a database called the Antibody Registry. And Leo Pascal, who's actually here, worked on an earlier version of this particular database. So what happened here is we took all of the data that we possibly could find about antibodies. We looked from about 250 different antibody companies. We worked with over 4,000 labs to register their reagents that they made. We gave all of the antibody reagents a persistent unique identifier. How many people have heard of PIDs and how cool they are? No one. Okay. Well, persistent unique identifiers, like kind of pictured here, you've seen them in the grocery store, right? These are not magic. They're just a way to get to a particular product or a set of information. And they've standardized the way that grocery stores work. They've standardized the way that Amazon works. They've standardized the way that lots of things work. But this is research, and research resists all standardization. So we try, we keep trying to get people to actually use these things in their scientific papers so that we don't have to guess. We don't have to create fancy AI to guess which product was actually being used in a particular study. So the scale of the problem is fairly large because there's about 150,000 antibodies papers every single year. And by the way, about 50% of these antibodies are actually bad or they don't work for that application. So half of these 150,000 papers are probably bad. And the kinds of questions that we were asking at that point is, which reagents are being used in this paper? Which other papers are actually using the same reagent? And how many antibodies are actually out there for people to use? So we did all of this in order to start to answer that question. By the way, authors can get a temporary RID in about 15 seconds. So we tried to make it as easy as possible for people to actually use these things. So once this data was gathered, maintenance started to become an issue. And the companies that we were working with needed to be convinced basically every time a new person would come in, which was really frequently, because companies like drop people and hire new people at the drop of a hat. And so in order to get the data, we knew we needed to have some kind of a contract between the company and our team. And then what we discovered to our dismay was that UCSD has no idea how to sign a contract with a company, or at least it takes them more than 6 months to do so, at which point the people we were talking to were already fired. So there was no way to do this, right, with UCSD. So what we did is we actually founded a company. And we founded a company in order to sign contracts with other companies. So we now have a bunch of contracts with companies that work with us to put their antibody data into the antibody registry, which is kind of cool. But there was no way to solve that problem within the university. So we had to go outside of the university to be able to do that. So currently, Antibody Registry, it provides these persistent identifiers for antibodies. Cool. It lists all of the reagents that are possible. So there's over 3 million antibody reagents in the antibody registry. We try to get authors, antibody companies, nonprofits, and others, and governments actually, to list all of their reagents. We have a whole lot of authors who are using this for antibodies. And at the moment, we have about 50 corporate partners in this. So this is the, the CEO hat. We keep going out. We ask antibody companies to list their stuff, pay us some money, sign a contract. They do all of those things. We can turn that contract around in under a day. It's fantastic. So here's a bunch of antibody companies. Some of them, like BioLegend, are local. Some of them, like Atlas Antibodies, are all the way in Sweden. But these are for-profits, nonprofits. We've got Thermo Fisher. We've got others. Okay. But this problem of not being able to find stuff in the scientific paper is a little bit bigger than that. So we knew that we couldn't find antibodies, right? But the pig people had problems finding pigs. Which pigs are being used in this study? Which flies are being used in this study? Which mice are being used in this study? Which cell lines are being used in this study? Which plasmids, right? And so what we did is we actually went across and we got data from about 25 different stock centers, major companies, nonprofits, for-profits, and we put everything together and we said, look, if we can go to the journals and say, look, guys, here are all of your reagents as far as we can figure them out. Can you get your authors to put these things into the papers? We'll give you one website with all of this data. Everything will be consistent so that the authors won't get confused. Can you try to do that for us instead of asking authors to go to each of these individual places, which might have data that's kind of all over the place? And so we did that. We created individual pages for every single research resource out there, they're all consistent. They have a piece of metadata that's associated with them. They have a name, they have a URI, they have a URL. They have ratings and alerts. We have a whole bunch of data that's associated with these things. We can track them within the literature. We can track them if they're near us and all this fun stuff. And so that asking the journals, right? We asked our friends who happen to run journals to come to the Commander's Palace at the Society for Neuroscience way back in 2012. We told them that there was a problem with reagents, and they didn't believe us. So 6 months later, the National Institutes of Health brought these people back in to a 2-day workshop that was sponsored by NIDA. And in that workshop, they were convinced that there was a problem, but there was no way they could possibly solve the problem of reagents in the scientific literature. And then we had a second meeting with a lot of those same people at the very next Society for Neuroscience, and they agreed to do a 3-month pilot project to ask their authors who are publishing with them to add RIDs to their papers. And so in 2014, we actually started this pilot project with 25 journals that would ask in different ways to provide RIDs. And so in just a couple of months over the holidays, we actually needed to create an entirely new website, integrate 25 different databases worth of data. We had to make everything, the search actually work, which was kind of a challenge. We had to make sure that everything was as consistent as it could be. And by the way, there was no funding for this, and we needed to have a help desk, which actually was really, really important for all of this stuff. That help desk was critical. We found out that only 2 journals started on time, and that was me nagging the second journal, which 15 days after they were supposed to start. So what I like to say is that we are currently in the 11th year of a 3-month pilot project, because it just so happens that no one really stopped doing the pilot once they started. And in fact, we got on online the journal Cell, eLife, Nature, Science, all of these big journals came on board between 2014 and now. And I think Science came on board the latest, and that was 2019. And then new smaller journals keep coming in. So we've had some impact, right? What we're asking people to do is to go to a website, search their favorite term, and then find this, cite this text and copy it, or there's a couple of ways to kind of have these little buttons that basically say, here's the thing that we want you to cite and how to do it. And then basically you take that, you copy it into the manuscript, and then when the paper is published, then that identifier, that code actually becomes part of the paper. That's usually in the methods section. And these IDs are actually reflected across different catalogs and different databases. So this is from Angene, this is from BioLegend down the hall or down the street. This is one of the larger mouse repositories, and they all have this little question that's like, hey, how do you cite this thing that I'm using? Here's how you can copy that citation to your clipboard. We had some fun when these RIDs came out into papers. We actually curate them automatically, which is very cool. So we had a tool that was extraordinarily simple. This is like 15 lines of Python walk into a bar with 2 API calls. This was much easier to do than creating an entire AI algorithm in order to find the answer to this very stupid question. Instead, we had a little bit of Python code, and it did a much better job. We had a student actually over the summer who created a Twitter bot that basically took the information that was being curated out and posted it to Twitter. And then later he created a Reddit bot and a Blue Sky bot. Basically does the same thing. It takes these papers and basically thanks authors for using RIDs. It's kind of cool. So did it all work? Like, what happened? So we have all these journals, big journals. Cell Press is a big journal. Nature is a big journal, right? And they're all doing this stuff. I'm sorry, these dates are not coming out very well. This is August 2016. This is 2016. This is 2018. But all these big journals actually do this. In fact, Cell Press has this entire method section, which has this reagent table. They did this with us. It was awesome. But did it work? So it turns out that journals, in order to enforce RIDs, they said, hey, that costs a lot of money, and we don't have money, so we're just going to do the bare minimum. So we said, well, what if you could actually get a robot to do that work for you, right? And we would charge you very little. So at this point, we actually did build that robot that NIH asked us to build, way back in 2008, because we do have the capability of doing so. But what happened is that we built this robot to actually interact with authors before they published the paper. So instead of, us finding it after it's published, we flag places in the paper for authors to actually interact with them and try and figure out how to improve the paper before it's published. And then when it's published, it gets to be a better paper with those RIDs and with a bunch of other things that the journals cared about putting in. So the journals cared about putting in things like the institutional review board statements. And the reason they care about that is because if you have patients in a study and you don't put that statement in, then the journal gets sued. And if the statement is there, then the university gets sued. So the journals really care about not being sued. So we put a bunch of stuff like that into the tool. And that, including things like, did you put your code up on GitHub? Did you put your data somewhere that's recognizable? Did you cite any clinical trials and a whole bunch of other stuff? And so we created this big tool, which of course included RIDs, but it also solved a couple of other problems for journals. So we now have a bunch of partner journals that are actually using the tool. So because we're a company, we were able to, again, sign contracts. And those contracts were with journals, which are nonprofit and for-profit companies. And we could turn those contracts around in under a day as well. Eat your heart out, UCSD. But We were also granted some very nice funding from the Chan Zuckerberg initiative in the company to work with bioRxiv to make some of this information also available there. And the question is, did it work? Well, I think the answer is yes. So we've been checking these RIDs and how many of them are actually being used in the scientific literature. Unsurprisingly, the cell journals that have that big table are leading the way, including, Cell Reports, which has over 100, or had at that point in 2022, 100,000 RIDs. And we had 500,000 RIDs in the literature in 2021 and before. And so we looked again a couple of years later, and there were over 1 million. So, yep, it's going. Things are definitely moving forward. We are getting a lot of people actually complying. This is what it looks like, more or less, when somebody does comply in a journal like eLife. They're actually links. And those links, we take those links and we drive traffic not to us necessarily, unless we have to, but we drive traffic to places like the Jackson Laboratory, which is where you get your mice. And the nice people at the Jackson Laboratories ask their authors to cite the mouse using the RID. And we drive traffic to them because these pages have a lot of information about that particular mouse that's being used in this paper. So we know that we can drive improvements in how people talk about their reagents and especially antibodies. So here, this is a paper from based on the first 100 papers that came out with RIDs in them. And you can see that the identifiability, the ability to find that antibody reagent goes from 50% all the way up to 90%, which is really incredibly cool. But in 2020, when I pulled this data, this is an overview of all papers. This is over 316,000 antibody papers. This is one of these big scale analyses where we used one of our AI robots to go after and process every, paper that was available for it to process. This is all the open access subset of the biomedical literature. 316,000 papers actually had a hit of an antibody. And in the 97, early 2000s, the number of catalog numbers associated with those reagents was like 10 to 15%. And you can see that this doesn't really change very quickly, that blue line, right? And then something strange happens. There's this inflection point. And this inflection point was actually us. This is the big journals like eLife and Cell that drove their authors to actually use RIDs. And then this inflection point in the overall literature is something that I would say probably is the pinnacle of my scientific career. I changed within the literature, our project changed how frequently antibodies are used reliably and not reliably by like 25% over about 3 or 4 years. That's incredible. And this has just continued to change, but this is from a paper that we published in 2020. And somewhat more recent work, we looked at how this is changing or continuing to change, and this is for mice. So this is identifiability again, and you can see that these orange bars, these are nicknames. So this is how people talk about animals, and this is, a nickname for an animal. A nickname means that it could be one of 2, one of 5, or 20 mice. So these things are not findable without asking the author. So when you look at 2011, half the papers are talking about animals like this. We get started in 2014. A couple of RIDs are in the papers in 2014. 2016, the big journals start to join. And then in 2017, both the big journals have joined, but also the place where people get these mice has also changed their website. And you can see that there's a big shift in RIDs. But the other thing that you're seeing is the percentage of these bad mentions where you can't find the animal, making these papers all not reproducible for a really stupid reason. This gets lower and lower and lower. And by 2022, this is under 10% of papers. So going again from 50 to 90% reproducible or findable, at least, which is really cool. So in terms of who I am a scientist that actually cares a lot about this really weird thing called reagents. I want them to be as nailed down as possible. I want to understand reagents in the scientific literature. I use a lot of tools at my disposal. I use AI, I use high performance computing, I use very large pipelines, and I use the simplest tools possible, like regular expressions. Those are my favorite tools in many cases. Where I can use those, I use those because they have almost no overhead. I also try to solve problems in probably different ways than you might be aware of. I can solve problems in a company that I can't solve in the university. And recently, because people don't trust companies, we've had to found a nonprofit. And we're going to start moving a lot of our stuff into the nonprofit foundation because that way we should be able to actually impact more of the scientific literature. So that's my story. I'm sticking with it. And thanks so much for listening.